We've all got this incredibly complex genetic blueprint, our DNA. But what happens when there's a typo in that blueprint? Today, we're going to crack the code on how scientists figure out if one tiny change in your DNA is just a harmless quirk or a serious red flag. So imagine this. You get a genetic test, and it flags one tiny change, a single letter typo in your 3 billion letter instruction manual. Is it nothing to worry about, or is it a warning sign for a potential disease? This one question is really at the heart of everything we do in clinical genomics. And as we're about to find out, getting to the answer is one heck of a scientific investigation. All right, so here's our roadmap for this genetic detective story. We'll start with that central question. Then we'll dive into the 5-tier systems scientists use to classify these changes. We're going to look at all the different kinds of evidence they have to gather, see how data from millions of people plays a huge role, and finally understand why the rulebook for all this is constantly being updated. Genetic testing is everywhere now, right? But the real challenge isn't just finding changes in our DNA, it's figuring out what they actually mean. That's where the real work begins. And this is the official term for it: variant interpretation. This is the crucial process of turning a raw string of A's, C's, T's, and G's into a clear, actionable answer for a doctor and their patient. It's the bridge from data to diagnosis. Now, to make sure every lab, everywhere, was speaking the same language, the scientific community had to create a standard rulebook. You can think of it as a shared framework for classifying every genetic person of interest they come across. And this is it, the 5-tier system that brings order to the chaos. On one end, you've got pathogenic variants. Those are the definite culprits. On the other end, benign, the innocent bystanders. But right there in the middle, you have the really tricky one, variant of uncertain significance, or VUS. This is the mystery that keeps geneticists up at night and can leave patients in a really tough spot. Okay, now what's really, really important here is that phrase, minimal criteria. These guidelines, which came out in 2015, they're not the final word. They're the starting line. They are the absolute baseline that all labs have to meet, and as we're going to see, this rulebook is always being refined as we learn more. So how does a variant actually get its classification? Well, it's not a gut feeling. It's a verdict. And it's only reached after weighing multiple, totally distinct lines of evidence, just like a detective building a case file. And this is the toolkit for our genetic detective. They're pulling in clues from massive population databases. They're using computer models to predict what a variant might do. They're running lab experiments to see what it actually does. And of course, they're looking at the patient's own story and their family history. Population data is one of the most powerful first clues. The logic is pretty simple, right? If a variant that's supposedly causing a rare disease is actually showing up all the time in thousands of perfectly healthy people, well, it's probably not the culprit. All right, this is where the lab coats come on. Scientists can actually take the variant into the lab to see what it does to the protein it's supposed to help build. Does it break it completely? Does it just slow it down? This is where we get hard biological proof, moving beyond just prediction to actual observation. And this brings the whole investigation right back to the human level. Did this genetic change just pop up out of nowhere in a child when neither of the healthy parents have it? I mean, that's a massive red flag. Or can you trace it through a family tree where everyone who has the disease also has the variant? These are the classic powerful clues of genetics. So let's zoom in for a second on just one of those tools, because the power of population data has been an absolute game changer for this entire field. I mean, just look at this number. Databases like Nomad have genetic data from over 800,000 people. This is staggering. It gives our genetic detectives this incredible power to see if a variant they find in a patient is a truly rare event or if it's just a common bit of human variation that's been around forever. And this chart just lays out the core logic so perfectly. If a variant's frequency in the healthy population is high, that first bar, it simply cannot be the cause of a disease that is, by definition, rare. The math just doesn't work. And that's really the key takeaway. This one piece of information, how common a variant is, can sometimes be powerful enough on its own to classify it as benign. And that saves a ton of time, money, and most importantly, it prevents a huge amount of unnecessary anxiety for patients. But here's the thing. That rule book we've been talking about, it isn't carved in stone. It has to be a living document, one that gets smarter and more precise as our own scientific knowledge grows. Let's just take one quick example. Back in 2015, the rule of thumb was pretty broad. If a variant showed up in more than 5% of the population, it was just too common. But now we're moving to a much more sophisticated model with precise gene-by-gene calculations. And that's because what's too common for a gene that causes a severe childhood disease is very different from what's too common for a gene linked to something much milder. It's all about tailoring the rules to the specific case. The entire approach is just getting smarter. Instead of a simple checklist, where evidence is either met or not met, the field is moving to a points system. This lets scientists weigh different pieces of evidence with much more nuance. It's especially useful when you have conflicting clues, some pointing toward guilty and some pointing toward innocent. This system lets the strongest evidence win out. And you can see that clear path of constant improvement right here on this timeline. You start with the foundational guidelines in 2015. Then you have expert panels refining them for specific genes. And now we're expecting a major update soon that's going to make the whole process even more data-driven and precise. So at the end of the day, why does all this matter? Because this constant evolution, this rigor, it directly improves people's lives. It means fewer variants get misclassified. It means you'll get a more consistent answer no matter which lab does your test. And critically, it helps us solve more and more of those uncertain VUS results, turning them into clear answers. This is what drives diagnostic accuracy and truly powers the future of personalized medicine. And this is the question that really drives the whole field forward. This rigorous, ever-improving process is how we turn mountains of raw data into life-changing knowledge. It really does promise a future where our own DNA holds far fewer secrets and a whole lot more answers.